Good morning, students. I'm Sirisha, PGT Botany, KGBV, Buchaipeta, Vizag District. Today we are going to learn the unit Diversity in the Living World, and in this unit we are going to learn our brief introduction about the living world, because it is already 30% reduced syllabus. We are going a very brief introduction about the first chapter, the living world, and in detail we are going to learn the biological classification. The living world. This is the first chapter in the diversity in the living world. In the world there are all living and non-living things. And what is the difference between the living and the non-living? Living means which have life, and non-living means which are not having life. And here in the living, we know the main characteristics are the growth and reproduction, whereas in non-living we does not see any growth or reproduction. But here, and in this not only growth and reproduction, they non-living also have the ability to sense, and also they have the interactions and metabolic reactions going inside. So here the growth is actually not only confined only to the living, but it also confined to the non-living. How non-living is? In the non-livings like mountains or boulders, the accumulation of the material is on that. So the growth also increases. Recently we have seen that Mount Everest is also the height is little bit increased. This is nothing but the accumulation of the material on it. And here, and but reproduction, metabolism, and interactions, and sensing ability, these are confined to the living substances. Like here, this is the reproduction. We cannot say that all living organisms can reproduce. There are some organisms that cannot reproduce, like a mule, infertile females, and etc. All these cannot reproduce. And not only for so here we cannot say that all living organisms can reproduce. And this reproduction means the giving rise to the progeny. This is we can see only in the living organisms, but not in the non-living organisms. And coming to here, the metabolism reactions required for the growth, all the biomolecules which are present in the humans or in the living organisms, these constitute the growth, and the biochemicals which are present constitute the metabolic reactions, and this is called the metabolism. And coming to the ability to sense, only living organisms can have the ability to sense. They can smell, they can taste, they can hear, and they can see. All these characteristics are only present in the living organisms. And coming to the interaction, or the interaction is also we can see only in the living organisms, but not in the non-living organisms. Here, so we know that all the living have growth, reproduction, metabolism, interaction, and sensing ability. And now how we know about so many in this living world, we know so many organisms are there. In this living organisms, they are like animals, birds, fishes, animals, so many are there. All these constitute the biodiversity, that is the number and the types of different living organisms constitute the biodiversity. So we know in our daily life, every day some new organism is we are finding out. Means some discoveries going daily. Some new organisms we are finding daily. That is, so many vast number of organisms or living organisms are present in the world. So how we can name them, or how we can identify, or what we can call them, becoming a confusion, because it depends upon the area wise and the region wise. They speak in different languages. So all over world we have so many languages speaking. So in this these organisms, these living organisms are named in with different names. So to avoid this confusion, ICBN, International Code of Botanical Nomenclature, this one has made a set of rules to name a particular organism, and this name it should be used all over the world. That is called as the nomenclature. So for the identifying the new organisms and naming them is nothing but the nomenclature. And okay, after naming them, we should make them into categories. That is, we should make them into groups, like plants as one group and animals as one group and fungi as another group. Like that, we have to group, or we have to categorize them. And the word used for the categorization is the taxon. So all these living organisms, they should be grouped. Easily observed characters are grouped under categories. Each group belongs to one particular category, and the scientific term for the categories is called taxon. The taxon is the unit of classification. Organisms represent taxon at different levels, that is, it is present in the form of hierarchy. Here, that is, the categories are differentiated from higher to lower. How? The first, they are grouped as a kingdom. Then they are divided. Then that is division, and division is for plants, and phylum is for animals. Next comes the class, and then order. Next family, then genus, and then species. These are the common categories to for every living organism. Let us learn about these categories. Species, group of individual organisms with fundamental similarities. It is the basic unit of classification. Species is the basic unit of classification. Genus, it is aggregate of a closely related species. Family, group of related genera with less number of similarities. Order, assemblage of families with a few similar characters. And a class includes all related orders, and phylum or division includes all related classes. And kingdom, this is the highest category, and all plants with various divisions. Let us see few organisms with their taxonomic categories. Man, Homo sapiens. Homo will be the generic name, and sapiens will be the species name. Homo is the genus, and Hominidae is the family. Primata is the order, Mammalia is the class, and Chordata is the division. This is how these again some housefly, mango, wheat, all these examples are given, and they are categorized like this. So here the mango, if you take a plant example, Mangifera indica, Mangifera will be the generic name or a genus, and indica will be the species name. And next, they will belong to the family Anacardiaceae, and the order Sapindales, and the class Dicotyledonidae, and the phylum Spermatophyta. This is how all organisms are categorized. Classifying the living organisms is the biological classification. Here, many scientists came forward to classify the living organisms. Aristotle was the earliest to attempt a more scientific basis for classification. He divided all living organisms into two groups. They are plants and animals. Plants are further divided into trees, shrubs, and herbs. Here plants are divided into again trees, shrubs, and herbs. So Aristotle, according to Aristotle, there are only two groups. There are plants and animals. And in plants, trees, shrubs, and herbs. And now in animals again they are divided into two groups. That is animals with RBC and animals without RBC. So examples for animals with RBC are mammals, lizards, birds, fishes, and animals without RBC are the insects, jellyfishes, etc. This is the Aristotle type of classification. Later, Linnaeus made two kingdom system of classification. Two kingdom system of classification include Plantae and Animalia. Plants are included under Plantae and animals are included under Animalia. So two kingdom classification actually it did not distinguish between the eukaryotes and the prokaryotes, and also unicellular and multicellular organisms, and photosynthetic and non-photosynthetic organisms. And in plants, bacteria, algae, fungi, bryophytes, pteridophytes, gymnosperms and angiosperms are included under plants in the two kingdom system of classification. This is even Linnaeus also proposed two kingdom classification, and in the plants he included bacteria, algae, fungi, bryophyta, pteridophyta, gymnosperms and angiosperms, and all animals under Animalia. But he was not able to distinguish between the eukaryotes and the prokaryotes, and the unicellular and the multicellular, and the photosynthetic and non-photosynthetic organisms. And organisms are grouped under one category even though they widely differ in other characters. So even though they are differing to each other, they all are grouped under one category. Prokaryotic bacteria are included under blue-green algae. Similarly, two kingdom classifications included all unicellular and multicellular organisms under one group. For example, Chlamydomonas and Spirogyra were placed under algae. The classification did not differentiate between the heterotrophic group, that is fungi, and the autotrophic green plants. So here all these are included under the plants, so he is not able to differentiate the fungi among them. So considering these above reasons, the two kingdom classification found inadequate for classification of organisms. After two kingdom system of classification, scientists felt that other characters are essential, like cell structure, nature of cell wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships. R.H. Whittaker 1969 proposed five kingdom classification: Monera, Protista, Fungi, Plantae, Animalia. We are now following the R.H. Whittaker five kingdom of classification. And coming to the characters, these are classified into five kingdoms, like cell type. For cell type, in the Monera, all are prokaryotic, and Protista, Fungi, Plantae, Animalia, all are eukaryotic. And coming to the character cell wall, in the Monera kingdom, they are non-cellulosic, and in Protista, they are cellulosic to some extent, some organisms, and in fungi they are not with the cellulose, they are with chitin, and in Plantae they are again cellulose, and in Animalia the cellulosic wall is absent, totally the cell wall is absent. And coming to the nuclear membrane, the nuclear membrane is absent in Monera kingdom and present in all other kingdoms. And coming to the body organization, it is cellular in the Monera and also cellular in the Protista, but multicellular or a loose tissue like mycelium in fungi, and it is tissues and organs in case of Plantae and Animalia. And coming to the mode of nutrition, it is autotrophic, that is chemosynthetic or photosynthetic, and also heterotrophic, that is saprophytic or parasitic in case of Monera. And coming to Protista, they are all autotrophic only, and in coming to the fungi, they are heterotrophic, that is saprophytic or parasitic, and in Plantae they are all autotrophic, that is they are photosynthetic, and in Animalia they are heterotrophic, and the mode of nutrition is holozoic or saprophytic, etc. Monera kingdom, this is the first kingdom in the five kingdom classification. All the prokaryotes included in the Monera kingdom. All prokaryotes like Archaebacteria, Eubacteria, Mycoplasma and Actinomycetes are included under the kingdom Monera. First we are going to learn about the Archaebacteria. These are the special monerans since they live in some of the most harsh habitats, that is extreme salty areas which are called as halophiles, or extreme hot springs that are called as thermoacidophiles, or extreme marshy areas which are called as the methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure. The cell wall does not contain peptidoglycan, but it contains pseudomurine. So here the cell wall consists of pseudomurine. Methanogens is also present in the gut of the several ruminant animals such as cows and buffaloes. Methanogens produce methane or biogas. After archaebacteria, next is eubacteria. Bacteria are the most abundant microorganisms. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans. Many of these bacteria live in or on other organisms as parasites, and some of them are as symbionts. Bacteria are grouped under 4 categories based on their shapes: spherical, that is you call as coccus, rod shaped, that is a bacillus, and comma shaped, that is vibrio, and spiral shaped, that is spirula. And here this is the diagram which is showing the bacteria can occur in different shapes. Bacteria are characterized by the presence of a rigid cell wall consists of peptidoglycan, also called as murine or mucopeptide. Before archaebacteria we have seen it is a pseudomurine, but here it is a murine or mucopeptide, and which consists of peptidoglycan. The plasma membrane is the phospholipid membrane, also contains proteins and polysaccharides. It is a selectively permeable respiratory enzymes that are associated with the cell membrane. The infoldings of the cell membrane is called mesosomes. In bacteria, the genetic material is basically naked. Cell organelles are not found in bacteria and in all prokaryotic cells except 70S ribosomes are present. So in bacteria there are no cell organelles but 70S ribosomes are present. Motile bacteria contain 1 or 2 flagella. Some are autotrophic and many are heterotrophic. Bacteria reproduce by binary fission. They produce endospores during unfavorable conditions. These are few characteristics of bacteria. Next is the cyanobacteria. They are also called as the blue-green algae. These are photosynthetic autotrophs. These are unicellular, colonial or filamentous, aquatic or terrestrial algae. Chlorophyll A is present similar to green plants. They are the most primitive organisms showing oxygenic photosynthesis. In this bacteria they are showing oxygenic photosynthesis because it is a pigment. Chlorophyll A is present in this. The protoplasm of the cyanobacterial cell is differentiated into the central colorless centroplasm surrounded by a peripheral pigmented chromatoplasm. So here the bacteria inside it is there is a protoplasm called as the centroplasm, and the peripheral it is the pigmented chromatoplasm is there. The red color of Red Sea is due to the Trichodesmium erythraeum is present in it. The colonies and trichomes or filaments are generally surrounded by gelatinous sheath. Here they form water blooms in the sea water. Nitrogen is fixed in the specialized cells called heterocysts. Example, nostoc and anabina. Asexual spores are Harmogonia. They are fragments or thick-walled akineids. Flagella are absent in the vegetative or reproductive phases. Here you can see the gelatinous sheath is surrounded to the whole body. That is why it is slimy in structure, and you can see the heterocysts and the akineids. And also the vegetative. So the difference between the vegetative cells, the akineids and also the heterocysts. Heterocysts, in this heterocysts the nitrogen is fixed in case of nostoc and anabina. And these are the akineids. These are the asexual spores which are formed during the reproductive time. And flagella are absent in vegetative or reproductive phase. There are no flagella in this group. And next about the chemosynthetic autotrophic bacteria. Oxidize various inorganic substances such as nitrates, nitrites and ammonia and use the released energy for the energy production. Here chemosynthetic autotrophic bacteria, they are used for the energy production. It plays a important role in the recycling of nutrients like nitrogen, phosphorus, iron and sulfur. Majority of the heterotrophic bacteria are saprophytes or decomposers. Bacteria cause insignificant impact on human affairs. That is, they are very useful and also harmful to the humans. Making curd from milk, production of antibiotics, fixing of nitrogen in legume plants, etc. are some uses of these bacteria. So from bacteria we are having the humans are benefited in some uses like making curd from milk, production of antibiotics, fixing of nitrogen in legume plants, etc. And they also cause harm to the humans, like cause damage to the human beings like crops, farm animals and pets. Causative agents of many diseases such as cholera, typhoid, tetanus, citrus canker in plants. And reproduction is mainly by fission, and in unfavorable conditions the bacteria produce endospores. Sexual reproduction is by primitive type and it is by the DNA transfer from one bacterium to the another bacterium. And the next group which is included under the Monera is the Mycoplasmas or the PPLOs. These lack cell wall. They are the smallest living cells and are pleomorphic. Pleomorphic is they have many shapes or that's many shapes is called as the pleomorphic. The shape may vary. They can survive without oxygen. Diseases caused by mycoplasmas are in plants which is broom, in cattle pleuropneumonia and in humans mycoplasmal urethritis. You can see the mycoplasma diagram in the picture. There is no cell wall. So here these bacteria does not have any cell wall. These are very small and it is surrounded by a lipoproteinaceous wall, and inside the genetic material RNA is present, genetic material DNA is present, and some RNA ribosomes and are also present. And next these mycoplasmas causes diseases, and these three are very important. In plants they cause the mycoplasmas cause which is broom disease, and in cattle it causes pleuropneumonia, and in humans it causes mycoplasmal urethritis. And coming to the next actinomyces in Monera, these are actinomyces are branched filamentous bacteria forming radiating colonies in the culture. Cell wall contains mycolic acid. Here they have cell wall, but it is a mycolic acid. Mostly they are saprophytic decomposers. So they are saprophytic, depends on other organism, and it is that they are saprophytic decomposers. They depend on the dead and decaying organic matter, and some are parasites like mycobacterium and corynebacterium. In streptomyces antibiotics are produced. That is, in streptomyces we get antibiotics. After we learn the Monera kingdom, let us learn Protista. This is the second kingdom of the 5 kingdom classification. Protista cells are single-cell eukaryotic with a well-defined nucleus and membrane-bound organelles. It acts as the connecting link between the Monera and the complex multicellular 3 kingdoms that are fungi, plantae and animalia. Boundaries of this kingdom is not well defined. Reproduction is by fission or by the formation of spores. Sexual reproduction is by the process involving cell fusion and zygote formation. Protista include the following members: chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. The first members which are included under the Protista is the chrysophytes. It includes diatoms and golden algae, which are also called as the desmids. You can see in the picture. These are found in the freshwater and marine water environments. These are microscopic and float passively in water currents and also called as the planktons. Most of the chrysophytes are photosynthetic. In diatoms, the cell walls form two thin overlapping shells, epitheca or hypotheca. The walls are embedded with silica and hence these cell walls are indestructible. So they form an accumulation of the silica on the cell walls of diatoms referred to as the diatomaceous earth or kieselguhr. Diatoms are the chief producers in the oceans. The soil of the diatomaceous earth being used in polishing, filtration of oils and syrups. So here the chrysophytes, these are available in the oceans and they float on the water, and they are very useful for us for polishing and filtration of oils and the syrups. The structure you can see in the diagram. This is the epitheca over the hypotheca, two cup like a soap box. It is present. Okay, the outer one is the epitheca and the lower one is the hypotheca. And these diatoms are generally referred to as the diatomaceous earth or kieselguhr because the presence of silica on the cell wall. The cell wall is made up of the silica. The next member is the dinoflagellates. The name indicates there are two flagella, and dinoflagellates are mostly marine and photosynthetic. Depending on the main pigments present in the cells of dinoflagellates, they appear in different colors. So dinoflagellates, they may vary in color. They may be green, they may be yellow, brown, blue or red in color. The cells of dinoflagellates have thick cellulosic plates on the outer surface. So you can see in the diagram, the body is covered with the cellulosic plates. See in the diagram, and here in this diagram you can see the flagella. Most of them have two flagella. One lies longitudinally and other lies transversely. You can see in the diagram the two flagella which is lying one in transversely and other in the longitudinally. And flagella produces spinning movements, and so they are called as the whirling whips. So this is also called as the whirling whips. And in these members, nucleus has condensed chromosomes even in interphase, and the chromosomes do not have histones, and such type of chromosomes is called mesokaryon. And the nucleus which is not having the chromosomes and which are not having the histones, and such type of chromosomes are called as the mesokaryon. Some of the marine dinoflagellates like Noctulica show bioluminescence. See in the picture, in the top picture, you can see the bioluminescence effect of the Noctulica. Here it's showing like a glowing effect in the sea. So next, the red dinoflagellates undergo multiplication that they make the sea appear in red. If you can see in the diagram, the sea is looking like a red color. See, the red color is due to the red dinoflagellates. The toxins released by large numbers of dinoflagellates kill other marine animals such as fishes. So these dinoflagellates release some toxins which are harmful to the other living organisms, other animals which are present in the sea. The next members in the Protista is the euglenoids. Majority of them are freshwater organisms. Before too we have seen they are marine, but here these members are freshwater organisms. The cell walls of euglenoids have protein-rich layer called pellicle, which makes the body slippery. Here the cell wall, it is made up of pellicle. Okay, they have in dinoflagella it is made up of cellulosic plates, and here it is made up of pellicle. They have two flagella, one is short and other is long one. Here also two flagella are present, but one which is lying inside the mouth and other which is coming outside. And here the anterior part of the cell bears invagination consisting of a cytostomic cell mouth. You can see in the diagram the cell mouth is present, the cytopharynx, the gullet and the reservoir. It forms a reservoir here. This is the cytopharynx. It consists of photosynthetic stigma or eye spot on the membrane of the reservoir. It is having one spot near that reservoir. You can see that is called as the eye spot or the stigma. Pigments of euglenoids are identical with those of higher plants. Euglenoids are photosynthetic in the presence of sunlight and heterotrophs in the absence of light. So if their sunlight is present, they will be autotrophic, that is because they have the pigments, photosynthetic pigments, they are autotrophs, but they behave as heterotrophs if there is no light. The reproduction is by the longitudinal binary fission and the palmella stage is found in the euglena. So reproduction is by the longitudinal binary fission and palmella stage is also seen in the euglena. We have seen the three members, chrysophytes, dinoflagellates, euglenoids, and the next members is the slime molds. These are saprophytic protists. Multinucleated mass of protoplasm is surrounded by a plasma membrane like plasmodium. Under favorable conditions, slime molds form an aggregation called plasmodium. So here you can see in the diagram, they are actually forming a round mass-like structure that is called as the plasmodium. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies which bears spores at their tips. In the below diagram, you can see small fruiting bodies. The black ones are the small fruiting bodies. During unfavorable conditions, these are formed. And these spores possess true walls and extremely resistant and survive for many years. They can survive for many years and these spores are dispersed by the air currents. Spores survive under adverse conditions. That is, they can survive in the adverse conditions and once the favorable conditions come, the spores again reproduce. In slime molds, the body moves along the decaying twigs and engulfing the organic material. This is all about the slime molds. And next coming to the protozoans members. Here these protozoans are also included under the Protista. Here all protozoans are heterotrophs. That is, they are not autotrophic, they are dependent. So that is, they live as predators or the parasites. So here the protozoans are not autotrophs, all are heterotrophs. Protozoans are believed to be the primitive relative of animals. Earlier it is included under Animalia, but now protozoans are included under the Protista. And these organisms do not contain cell wall. They do not have cell wall and the protoplasm is surrounded by the plasma membrane. So here cell wall is absent, but the cell membrane, that is the plasma membrane, is present. Four major groups of protozoans are amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. So here the protozoans are again classified into amoeboid protozoans, flagellated protozoans, ciliated protozoans and sporozoans. This is based on the type of locomotion, that a movement, the amoeboid movement, the flagella, how they move, the locomotion, based on the locomotion habit, these are grouped, the protozoans are grouped. So coming to the first one, the amoeboid protozoans. Amoeboid protozoans, organisms live in fresh water, seawater or moist soil. These protozoans use false feet, that is pseudopodia, to capture the prey. Some are parasites. Everyone know about the amoeba. These are the pseudopodia which used for the locomotion and also to capture the food. And some amoeboid protozoans act as parasites also. Next protozoans are the flagellated protozoans. Here, organisms under this group are either free living or parasitic. The parasitic forms cause diseases. Diseases such as sleeping sickness caused by trypanosoma. You can see in the diagram the Trypanosoma gambiense. This is a parasite which lives in the humans and it causes the sleeping sickness. And next is the ciliated protozoans. It is here in the flagellated protozoans, we are seeing a free flagella over here. With the help of this, it can move easily. And next comes the ciliated protozoans. These contain cilia. With the help of the cilia only they can move. Actively moving organisms because of the presence of thousands of cilia. Coordinated movement of rows of cilia causes the water laden with food that passes inside the gullet. Example, paramecium. In the paramecium diagram you can see the cilia, how they are present all over the body, and you can see the gullet in which the movement which is it helps in the movement, and this is all about the ciliated protozoans. And coming to the sporozoans, this includes the diverse organisms that have an infectious spore-like stage in their life cycle. That is, example, Plasmodium, the malarial parasite which is causes malaria. Here the Plasmodium, this is a sporozoan, and it is a protozoa which is it causes malaria in the humans. These form spores during their life cycle. Note down the questions in this chapter. What is the basic unit of classification? Define it. This is the species, you have to define that. And give the scientific name of mango. Identify the generic name and the specific epithet. You have to write the mango scientific name, that is Mangifera indica, and you have to write the Mangifera as the genus name, and the indica is the species name. This is what you have to write in the very short answers. And coming to the short answer questions, what are the characteristic features of euglenids? It is the most important question which is asked several times in the papers. You have to write all the characters of euglena, and you have to draw the diagram of the euglena, and you can give all the characteristic features of that. It is present, the pellicle is present, and it has two flagella. All these you can write about the eye stigma, eye spot or stigma, and about all remaining characters also you can write. And give the salient features and importance of chrysophytes. You can write about this chrysophytes, the diatoms. About this we have discussed the importance about the polishing and all these you have to write. This is about the assessment. You please learn once again, go through the Monera kingdom and also the Protista kingdom, and these questions, some important questions are there. Very short answer questions definitely come from the Monera kingdom and the Protista kingdom. In the next session we are going to learn about the kingdom fungi, and also about the viruses, viroids and prions, and a small information about the Plantae kingdom and also the Animalia kingdom. That's all for this session. You better go through again the two kingdoms in the 5 kingdom classification, that is Monera and the Protista, and in the next session we are going to deal with the fungi. And thank you students. Hope you understood the lesson.